speakers today. We have Dr. Laura Vandenberg from UMass Amherst and Dr. Cal Baier-Anderson from the U.S. Environmental Protection Agency. Dr. Vandenberg is going to speak first, followed by Dr. Baier-Anderson, and I'm going to introduce both of them, read brief bios now, so that we can kind of move seamlessly through the presentation. So first, Dr. Laura Vandenberg is an assistant professor of environmental health at the University of Massachusetts at Amherst School of Public Health and Health Sciences. And she has a Ph.D. from Tufts University School of Medicine. Her work focuses on how low-level exposures to environmental chemicals can lead to diseases, including breast cancer. And her work also critically evaluates issues that affect risk and hazard assessments for endocrine disruptors, including low-dose effects, non-monotonic dose responses, critical windows of susceptibility, and routes of exposure. Dr. Vandenberg is an author of more than 50 peer-reviewed papers, 5 book chapters, and has served on a number of U.S. and international expert panels to address endocrine-disrupting chemicals. Dr. Cal Baier-Anderson is a toxicologist with the U.S. EPA, as I said, Risk Assessment Division, where she conducts hazard, risk, and alternatives assessments on both new and existing chemicals. Prior to joining the Risk Assessment Division, she was a toxicologist in the EPA Design for the Environment program and a health scientist with the Environmental Defense Fund, as well as a part-time assistant professor in the Department of Epidemiology and Preventive Medicine at the University of Maryland, Baltimore. She's also worked as a risk assessment and risk communication consultant, and she earned her Ph.D. in toxicology from the University of Maryland. So as I said, we're going to start with Dr. Vandenberg, and I will make that happen right now here. Just one moment. Okay. So Laura, you should have, and yes, I can see your screen.
Great.
Thank you so much. Go right ahead.
Okay. Well, thank you for the invitation to speak to this this very multidisciplinary and interdisciplinary group. I'm an environmental health scientist. I'm actually really trained as a biologist, and so what I've been asked to do is to provide you with really a very broad overview of this group of chemicals known as endocrine disruptors, and try to explain why you should be worried about them, and then to more specifically focus on BPA, to sort of set you up for the second talk this afternoon on alternatives assessment for BPA that's been expertly performed by the EPA. So in 2010, a group of us reviewed every single paper that had been published looking at urine samples that had been collected by people around the world. And what we wanted to do is to see, was there some general consensus on whether BPA and its metabolites could be found in those urine samples. And each one of these red lines that you can see here is an individual study, and what percentage of the people in that study were found to have BPA or its metabolites in their urine. And what you can see is that overwhelmingly, when you look at all of these different studies, the vast majority of individuals have BPA and its metabolites in their urine. And there's 2 exceptions that are worth talking about. One is this study right here, where not a single person that was examined had BPA in their urine, suggesting that they weren't being exposed to BPA. But in fact, when you really dig in and look at that study, what it showed was that they were looking at BPA with such old technology that they couldn't detect it because they weren't looking at it properly in those urine samples. So the technology that we use to answer our questions is very important. And I think this study here also deserves some attention. This is a study that was done in rural China. And they found that about 45% of individuals in those rural Chinese villages had BPA or its metabolites in their urine. And a follow-up study looking at rural Chinese has shown that number is significantly increasing, suggesting that uses and exposures of human populations, even in fairly rural areas in developing countries, is actually increasing. When we looked at large reference populations, so thousands of individuals that were representative of whole countries, we found this interesting dynamic. So here you're looking at age groups of 6 to 11-year-olds. These are adolescents, and then these are the adults. And with these large reference populations, we saw this really interesting pattern forming where reference American populations had about twice as much BPA in their urine compared to Canadian populations. And to date, there's really no good explanation for why Americans are potentially being exposed to 2 or 3 times as much BPA as Canadians. And after we published that paper, it was one of the more interesting responses that I've received, because I got emails from all kinds of people up in Canada with their suggestions on why their exposures were lower, none of which actually bear out in terms of the use of BPA in consumer products. They use it in cans up there. We use it in cans down here. It's human actual uses of those products, so there's no evidence that Canadians eat less canned food or handle thermal paper any differently than Americans. So this is a really interesting pattern that deserves some additional attention. And as I've sort of alluded to, we know a lot, and our knowledge really has grown in the last decade about how BPA is being used in various consumer products, and therefore how humans are being exposed to it. So I think the plastics, reusable food storage containers and plastic water bottles are very widely known to contain BPA. And of course, not all plastics contain BPA. We're talking about polycarbonate plastic, which uses BPA as a building block. But a lot of other plastics contain low levels of BPA in them because it becomes a contaminant in the recycling stream. So plastics that are thought to be BPA-free, because they're not polycarbonate, the BPA was never purposefully added as an ingredient, actually can contain BPA as a contaminant. Food can linings has gotten a lot of attention, and risk agencies around the world have anticipated that the vast majority of human exposures to BPA are actually coming from food can linings, although ideas about exposures from other sources are shifting. And in the second talk this afternoon, you'll hear from Cal about BPA and its use in thermal papers. So just 2 sentences about this, since you'll hear a little bit more. BPA is added in its free form to thermal paper because it acts as a developer in that paper. Thermal paper, we're talking about the kind of paper that gets spit out at your ATM, at the gas station, or the gas pump itself, but also a lot of receipts that we get at the grocery store, at restaurants, are made out of thermal paper. And thermal paper works by having a chemical that's basically sprayed on the paper surface itself, and it changes color when heat is applied to it. And that paper, the chemical is not tightly bound to that paper, so we can absorb it through our skin by handling that paper. And then some other lesser known sources of BPA exposure. Some dental sealants are made with a material that if it's not completely polymerized, releases BPA. This is probably mostly a concern for human exposure right after the dental sealant or dental material has been applied to the teeth. There is plenty of evidence suggesting that medical equipment, including equipment used in the neonatal infant care units, as well as dialysis equipment, contains BPA. And of course, we if we're making a choice between a very vulnerable baby that's been born versus exposures to environmental chemicals, I think most of us would agree that that baby should live. But some studies are suggesting that there is medical equipment that has low or no levels of some of these environmental chemicals, including BPA. So there are already alternatives that are available. And then there's all kinds of weird exposures that we don't think about, including BPA is added to the filters of cigarettes. So if you need an excuse to quit smoking and the other nasty things in cigarettes aren't enough, then you can lower your BPA exposure by quitting smoking. We know that consumer activities and also manufacturer activities can really influence human exposures. So this was a study that was done at Harvard by Jenny Carwile's group, where they asked students to consume all of their cold beverages in either a container made out of polycarbonate, for a week, followed by a week out of a container that was not made out of polycarbonate, or vice versa. And what they showed was that the concentrations of BPA metabolites in urine were about twice as high when those individuals were using a drinking container made out of polycarbonate. So replacing your drinking container with one that's not made out of polycarbonate potentially could reduce your exposure. And then there's also, this is an example of a manufacturing response. This is a study that was done in Japan, college students, and they consume coffee and tea in cans the way that we in America typically consume soda in cans. And they asked the individuals what kind of consumption they have. So on left in red are individuals that reported fairly low consumption of these coffee and tea canned drinks. And in the purplish color are individuals who reported consuming more of them. And as you can see, individuals who reported consuming more of these drinks had higher levels of BPA metabolites in their urine. Now, the manufacturers changed the composition of the can coating, and a follow-up study showed that there was no longer a relationship between the number of those canned drinks consumed and BPA exposures. So a change in the can coating really did have a quite significant effect, especially on those individuals that consume a lot of those beverages. So why are we worried about BPA? Now, the story of BPA really started in the 1800s when it was first synthesized, and then in the 1930s when it was explored as a pharmaceutical estrogen. So in the 1930s, it was known to have estrogenic properties. And it was put back up on the chemical shelf, so to speak, when other chemicals like DES were found to be more potent synthetic estrogens. So this figure shows you while BPA and estradiol, the natural estrogen, don't look a lot alike, they do share a small number of structural similarities that allows BPA to bind to the estrogen receptor and to mimic estrogen, both in cells in a dish and in the bodies of creatures. So, and BPA, more explanations or more explorations have shown that it not only can bind to the estrogen receptor, it has a lot of other activities as well. And I'll talk about that in a second. If we're thinking about BPA as an endocrine disruptor, it really falls under just a tiny percentage of all of the endocrine disruptors that we are likely exposed to in our everyday lives. They come from a large variety of consumer products. And some of these also are naturally occurring. So soy contains phytoestrogens. So we're exposed to endocrine disruptors from natural products, as well as a lot of other kinds of products that we interact with in our everyday lives. As we've grown to understand more about endocrine disruptors, our knowledge of how something that can alter the actions of hormones has really been bolstered by the field of endocrinology, which for 100 years has been building a knowledge base describing what hormones do in the body and how they do that. And I think I'm showing you this figure here to try to impress upon you that the list of hormones, depending on whether you count neurotransmitters, which some neuroendocrinologists do and some don't as hormones, is a huge list. So really the number of hormones in our bodies goes far beyond the estrogens, androgens, and thyroid hormone that most of our scientific studies and assays are capable of really assessing. There's a lot more hormones in the body, and therefore a lot more things that we as endocrinologists, biologists, and toxicologists will have to do to understand endocrine disruptors. And just to make it clear, on a couple of my slides, I refer to endocrine disruptors as EDCs, which just means endocrine disrupting chemicals. We have to remember that hormones act at low doses, very, very low doses. I show you a picture of a swimming pool here because during development, androgens, the male hormones, are active in the part per trillion dose. So that's the equivalent of 1/20th of a drop of water in an Olympic-sized swimming pool. And in adults, our hormones are functioning in the part per billion concentrations, which is about a teaspoon in this Olympic-sized swimming pool. So when we're talking about environmental chemicals being present at low doses, that's not a reason to suggest that they won't be biologically active, because that's exactly in the range where hormones are acting. And we also know from studies of hormones that low doses can produce very potent effects. So shown here, the difference in hormone concentration between 0x and 1x is x, and that increase in hormone concentration increases the numbers of receptors that are bound by that hormone by 50%, versus in a higher range, the difference between 5x and 4x is still x, but the number of receptors that are bound increase is only about 4%. So this is a biological system that is primed to respond to very slight increases in the low range and to produce sort of amplified biological responses because of it. We also know that hormones act via receptors. So the sort of metaphor that's often used is the idea of a lock and a key. And so you can increase the response of a biological system by increasing the hormones that are there, the number of hormone molecules that are present, but you can also increase the response by altering the number of receptors that are present, or the location of those receptors that are present. So this is a biological system where both the lock and the key are very important. We also know that hormones can have what are called non-monotonic dose responses. So when we think about a traditional toxicant, think about formaldehyde or benzene, we normally expect that more of an exposure would cause more of an adverse outcome. So you're exposed to a little bit of formaldehyde, you might not feel good, but you're not going to die. Exposed to a lot of formaldehyde, you're going to have a really bad effect. Hormones don't act like that. So I mean, they can for some outcomes, but for many outcomes, they actually look more like this graph that I'm showing you here. And the reason why is that in the low range, hormones act like hormones. They can cause proliferation of cells, they can cause differentiation of tissues and organs, versus in the high dose range, hormones actually start to act like toxicants, so they can kill in the high dose range. And this has been shown for endocrine disruptors also. This is a study looking at the effects of PCBs and their influence on body weight. And what the author showed was that it's actually a moderate dose of many of these PCBs that causes a significant increase in body weight. Now, that doesn't mean that those high doses are better for you, because you're going to see different effects on different endpoints at those high doses. This has been shown, these non-monotonic curves have been shown in literally hundreds of studies of endocrine disruptors for cells in a dish, for animals in the lab, and there's about 40 studies that have shown these non-monotonic responses in human populations. What this means is that for hormones and also for endocrine disruptors, it is not necessarily a prudent thing to study high doses and try to extrapolate that information to what can be expected at low doses. Finally, I think this is the point that I find the most important because of my training as a developmental biologist, is the understanding that hormones do different things at different periods of development. A woman who takes ethinyl estradiol, that's birth control. She will not be fertile when she's taking that ethinyl estradiol, but when she stops taking it, her fertility will be regained. A fetus that is exposed to ethinyl estradiol will have the actual developmental trajectory of its cells affected and shifted in ways that are not reversible necessarily. And Theo Colborn, who passed away this past year, but is sort of the grandmother of the field of endocrine disruptors, said this quote: "From the day of conception until an individual is born or hatched, the development of each stage of life is fully under the control of hormones. Changes that happen during development are far less reversible than those occurring in an adult. You can't go back and rewire the brain." And so during development, which actually extends into the postnatal life for some of our organs, including our brain and reproductive system, when you're exposed will dictate what kind of outcome you expect to see. And some of these outcomes may not arise until later in life. So with rodents, we can see an early life exposure to many estrogens can lead to mammary cancers that arise in the adult period of those animals. We know that human disease trends for many endocrine-related diseases are on the rise. These are just two examples. The top is testicular cancer. The bottom is female breast cancer. And this has been shown for dozens of different endocrine-related conditions. So going back to BPA, and I'm going to wrap this up and send it over to Cal in just a few minutes. Why are we worried about BPA? Well, when a group of 309 chemicals were ranked according to their toxicological profile, BPA was ranked as number 3. And this is work that was done with scientists at the National Toxicology Program and the EPA. And BPA's ranking as number 3 for all of these chemicals was based on all of the different receptors that BPA can interact with, which are shown here in this thing that's called the tox pie chart. And animal studies, I'm not going to talk about these in too much depth, but there have been literally hundreds of studies that have shown effects of BPA on a wide range of different endpoints. And most of these effects have been seen following early life exposures and showing effects on different organs and organ systems and disease endpoints in later life. We in the lab I did my Ph.D. in and in my current lab, we have been studying the effects of BPA on the mammary gland with the idea that we know from other estrogens that early life exposure can lead to mammary cancer that develops in later life. These changes and alterations in disease endpoints have now been shown in the monkey, which is shown here, in the rat, including palpable tumors that have been shown in the rat. Some of these tumors are frank carcinomas, so actual cancers induced by BPA alone, as well as in the mouse, shown here and shown here. There are more than 100 epidemiology studies that have been performed on BPA where associations have been shown between BPA exposures and a range of human diseases. These studies, of course, have to be interpreted with extreme caution for a number of reasons. The main one being that we are all exposed to BPA, so there isn't an unexposed group that we can compare ourselves to. This is a challenge in environmental epidemiology, but epidemiologists have tools to tackle these kinds of challenges. The really great studies that arise from epidemiology studies typically look at early life exposures and late life diseases. You can imagine that those are studies that take literally decades to perform. One was just published last week looking at DDT and early life exposures to DDT, exposures in the womb, and breast cancer risk in those women. That study took more than 50 years to collect the data and analyze and publish. So similar studies with BPA you would expect to take several decades to perform. And BPA replacements, a major replacement for BPA has been bisphenol S. These are the structures, BPA structure and BPS's structure here. It's used in a lot of BPA-free plastics. The concern is, of course, that looking at these two molecules, you might predict, just based on their appearance and maybe their names, that they would have similar biological effects. BPS is much less studied than BPA, but the few studies that are available suggest that it also has endocrine disrupting properties, and it seems to be that most of its biological activity is limited to being an estrogen, whereas BPA has a lot of other activities. Unfortunately, replacing one endocrine disruptor with another is an example, and that there are others from the literature of a regrettable replacement. So we certainly do not want to encourage the phasing out of BPA and the phasing in of BPS, a less studied but equally or potentially equally concerning chemical. And there is work being done by a lot of different groups to change how we actually test chemicals to determine if they're endocrine disruptors. I think probably the take-home message from these various testing strategies is that without performing the right tests, we should not assume that something is safe, because as product manufacturers, as public health professionals, we certainly hope that the replacement of something we know nothing about wouldn't be seen as a better option. We actually have to know some information and decide what safe really means. And also, if we're going to test chemicals, let's test them before we produce the product, so that if we do achieve something that we would think of as a positive effect, meaning, yes, this is likely to be an endocrine disruptor, let's not develop it at all. Let's continue to look for a safer alternative. Now, that is a huge challenge, and that's where I'm going to pass this off to Cal, who's going to talk about that challenge of finding replacements for or alternatives for BPA, at least in some products.
Great, thank you so much, Laura. And let me make Cal the presenter. All right.
All right, I'm working on it here. Can you see my screen yet?
Yes.
Okay, let me get the slide show up. And all right.
Beautiful.
I think we're ready.
Thank you, Cal.
Thanks, and thanks, Laura. That was a great presentation and a nice setup for what I'm going to talk about. So just a few things to point out on this opening screen. First of all, I am now with the Risk Assessment Division, but formerly I was with the Design for the Environment program, and it was when I was in DFE that I led the partnership to evaluate the alternatives to BPA and thermal paper, and today I'll be presenting the results of that project, which concluded over a year ago. And let me point out that the views are mine alone and do not represent agency policy or endorsement. So the Design for the Environment program, even though it's located within a regulatory agency, it's actually a set of voluntary partnership programs, one of which is the Safer Choice Labeling Program, where we evaluate products that are made with chemicals, and we evaluate each chemical against criteria. If the chemicals meet our criteria, then the product can bear the Safer Choice logo as an incentive. Another program is the Chemical Alternatives Assessment Program. This is really important because the goal is to characterize the environmental and human health impacts of chemicals and their alternatives to promote that informed substitution that Laura had talked about. So many times there's a desire to move away from a chemical, but if you don't know what chemical substitute you're moving to, there may be unintended consequences. So the framework that has been developed by us and others is to promote this comparative examination of the hazards associated with chemicals and their alternatives in order to understand what might be safer, what trade-offs may be present, and make that informed decision. So in the Chemical Alternatives Assessment Program, we identify and evaluate alternatives. And DFE focuses on that comparative hazard assessment, although we do take into consideration exposure and use and other life cycle issues. That is very important to setting the context for the comparison. We also involve stakeholders from across the spectrum of interested parties, and so that includes chemical manufacturers, product manufacturers, retailers, environmental groups, unions, other representatives from other state and federal agencies. So we all come together to scope out the project, and which we find very helpful and informative, and it really leads to a much deeper understanding of the substitution context. So the outcome of the alternatives assessment is generally a report that compiles the best information we have on hazard from existing data. This may be toxicity testing as well as analog information. Those are structurally similar chemicals, as well as computer models. And the evaluation is basically a screening level which was developed based on the methods, the tools and methods and approaches we use to evaluate new chemicals that we're required to consider under the regulatory Toxic Substances Control Act. We do not rank or benchmark chemicals. Instead, we leave it in this compare and contrast format for others to use this information. And in addition to minimizing the potential for these unintended consequences, the information can be integrated into a company, generally product manufacturers, performance and cost analysis, and become just a routine part of their screening and evaluation process. So Design for the Environment considers a broad range of parameters when evaluating chemicals, and that includes both human health and environmental traits or endpoints. And you can see the long list here. I won't read every single one of them. I will note that the framework is flexible. It allows for additional criteria when they're relevant and available. One thing I will point out is for most of the for most of the endpoints, we have criteria that divides that evaluates chemicals and bins them into high, moderate, or low concern. And I'll show an example of that in a second. But for some endpoints, we don't really have the depth of knowledge and data available that allows us to parse the results of the hazard profile into this high, moderate, low bins. And endocrine activity is one of those endpoints. So instead what we do is describe the available data in the hazard profile, but we don't give it a ranking of high, moderate, low concern. So why we chose to evaluate BPA and thermal paper and alternatives for this use, it grew out of an action plan that EPA released in March 2010. which identified BPA as a potential concern. It tasked the Design for the Environment program to evaluate alternatives to BPA in thermal paper. So that was our task. The intent was that the report would help product manufacturers reduce the likelihood of unintended consequences of using substitutes for BPA. And the link is provided at the bottom of this slide. So we pulled together a partnership, as I alluded to previously, to help identify the alternatives to BPA in thermal paper and compile associated information regarding use, exposure, and life cycle considerations. So through this partnership, we identified 19 alternatives, which we evaluated based on the hazard criteria associated with BPA and the functional alternatives, and we've prepared these profiles for each chemical based on a review of the literature, any structure-activity relationship information or computer modeling, and in some cases proprietary information shared by stakeholders. And when we did receive proprietary information, we worked with the owners of that information to disclose as much information as possible to inform the development of the hazard profile. So as Laura mentioned, the BPA is used to make thermal paper. Thermal paper is a highly engineered paper. There's a base of paper and there's a slurry on top, and that slurry contains a dye, generally a leuco dye, which is white, and BPA and some other chemical additives. And under the presence of heat or pressure, that BPA interacts with the dye to change the color from white to either like a purplish or black color. So there's definitely unreacted BPA in the thermal paper. It's not polymerized. It's just present in that slurry that can rub off. And so we know that workers in certain occupations, such as cashiers and restaurant servers, may be at greater risk of exposure. Similarly, children may be may experience greater exposures due to hand-to-mouth behavior and mouthing of inappropriate objects, such as thermal paper. The recycling of thermal paper is also a concern because it can contribute residual BPA to the supply of recycled paper and may be an additional source of release to the environment. So I mentioned we have these hazard criteria that kind of bins chemical endpoints into high, moderate, or low concern, in some cases very high or very low concern. So the development of criteria is for DFE. We took into account criteria that's being used globally, such as the Globally Harmonized Criteria for Classification and Labeling, or GHS. We've also have criteria that is used in the Office of Pesticide Programs, as well as internally within my division, Risk Assessment Division, and we took into account all these different schemes for kind of ranking the level of concern associated with tox data for each endpoint and for different routes of exposure, and came up with this set of criteria that we apply to each chemical. And the criteria are fairly flexible because it allows us to use either measured data from toxicity studies or analog data or computer models. So this is just a snapshot. You can go to the full document, which explains the derivation of this data and more information on how to apply it, and that's the link at the lower left corner. We also have criteria for environmental toxicity and fate. As you can see here, again, the similar idea is you bin it based on high, moderate, low, depending on the effect level that you see in the toxicological study or the environmental persistence or bioaccumulation study. So it allows us to compare and contrast the hazard profiles and the environmental toxicity and fate of chemicals across the board. So we have actually 3 ways of displaying the outcome of the alternatives assessment report, and this is the kind of the highest level summary way to do this. And so here you see a number of chemicals. These happen to be, BPA is at the top of the table, and chemicals that are structurally similar to BPA that were included in the alternatives assessment are all shown on this page. A couple things to point out. First of all, if the endpoint, the human health effect or aquatic toxicity or environmental fate effect is based on measured data from a study, then you see the results in color, either green, yellow, or red. If it's based on analog data, a structurally similar chemical or computer model, then the outcome of the hazard evaluation is in italics, black italics. So a couple things you can see. Yes, we have quite a bit of data for bisphenol A, but for most of the other alternatives, we have very little data. So often BPA was the analog that we used to assess the hazards of the alternatives because we had no other data to do this. On slide 11, we see more BPA alternatives, although these are grouped because they are structurally similar to bisphenol S or BPS. And BPS is at the top line of this table, and the structurally similar alternatives are below it. And as Laura pointed out, we have a bit more data for bisphenol S, but it's not quite as well studied. We don't have, for example, a 2-year cancer bioassay for bisphenol S, and so that was evaluated based on analog and expert judgment. So again, you know, we have some data, but when you don't have data, you have to fill that in with analogs, computer models, and expert judgment. That introduces some uncertainty. And then for the third slide, depicting the results, these are BPA alternatives, but they're structurally distinct. So they're not similar to BPA, they're not similar to BPS, they're a little different. They're a little harder to evaluate also because, again, they're data poor, so there are lots of data gaps. But this, again, this allows us to compare and contrast not only like the level of hazard associated with each endpoint, but where the data gaps are and the uncertainties are. Now, another way of presenting this information, and this is just an example of the hazard profile because there's a lot of detail buried in this report, and at the bottom of this slide you see the link to this report. So with the example of bisphenol S, we show the structure. We have a lot of information about the physical chemistry of this chemical, synonyms, metabolites, degradants, transformation products, if relevant, analogs, etc. So there are a lot of details buried in this report. We also include just very brief descriptions of each study that we considered when developing the hazard profile. And then you can see in the purple blurb in the middle, there's a summary of the justification of our hazard ranking for that endpoint. In this case, acute mammalian toxicity, low concern because of the results of a number of studies that are depicted below it. So just some observations from the outcome of this project, that most chemicals suggested for inclusion were structurally similar to BPA or BPS, and many chemicals had significant data gaps. So that makes it more difficult to evaluate and anticipate trade-offs that might be associated with these alternatives. It can be difficult to compare chemicals that exhibit different toxic effects, the apples and oranges situation. So in addition to endocrine disruption, which Laura described for BPA, there are a number of other hazards that need to be considered as well, and so it's very difficult to kind of summarize and come to conclusions when you see so many different effects in so many different chemicals. But hazard trade-offs are common among alternatives. It's very rare that you see a chemical that has no hazards associated with it. Chemicals are really complicated. So some other options to consider in addition to, in addition to chemical substitution is redesign of thermal paper, different types of printers, and e-receipts. I do want to point out, I'll try and wrap this up now, that it's, developing hazard criteria is not an easy thing to do, and although it's very handy to have criteria that can be used to compare the chemical hazards, but there are, a lot of the emerging concerns such as endocrine disruption or effects on wildlife. Most chemicals lack data for these endpoints, and there's an absence of consensus on hazard ranking, which limits our ability to derive hazard criteria to employ this in the alternatives assessment framework. But I'm sure that's coming. So one other final point on alternatives assessments is a number of people are thinking strategically about how we can systematically integrate a broader range of considerations and trade-offs into the alternatives assessment framework, such as differential exposure and life cycle considerations. So this is actually an exciting field, an interesting field to be in. So for more information, I'll leave you with these links and my email. So thanks.
Great, thank you so much, Cal. That was fantastic. Okay, well, let's see. We've got about 10 minutes for questions and answers. I think I will just sort of put this up as a placeholder here. So again, there's a question box. If people have questions and would like to type them in, there is also an ability through the control panel to raise your hand in a virtual manner, and if people would like to do that, I can certainly selectively unmute people and allow you to ask questions yourselves, if that would be easier, and we can make that work. So let's see. Sorry, as I try and just make sure I can see this. So here's a question, and this is a question I think to either one of you. What would you recommend as a best first step policy to help move the market away from the use of BPA and thermal papers?
Well, I don't really do policy work, so, and I don't know if Cal does, so maybe she's the better person to address. I think that the phasing out of chemicals that are known or suspected endocrine disruptors from a wide range of consumer products is a place to start. So that to me seems to be sort of a legislative thing. I don't think that's something that the EPA can do on their own without action from Congress, and I'm not sure that that would happen. But I do know that there are companies that are deciding not to use BPA or anything else that has at least estrogenic properties in consumer products like thermal paper.
Okay, so I'll jump in then. Thanks, Laura, for starting that response. I think that this is my personal view. It requires a multi-pronged response because there are a lot of untested chemicals, so there are a lot of data gaps and unknowns. So it's really hard to kind of guarantee that you're moving to a chemical that is safer. But I think we need to try. And I think asking the questions for product manufacturers, asking the questions, that's a good first step. So on the one hand, we need to continue to engage the chemical designers, so companies that are developing new chemicals, in this conversation about what constitutes safer and how we demonstrate it. Laura pointed to some testing regimes that are being developed that will be useful for that purpose. The other thing is with thermal paper specifically, we have to remember that's a highly engineered paper. So it requires the use of multiple chemicals in the development of thermal paper. And just anecdotally, a woman called me. She said she had a case of leftover thermal paper, and she wanted to give it to the church preschool so that they could color on the paper. And did I know if it had BPA in it? My response was, you know, whether it has, I don't know if it has BPA in it, but it has a lot of chemicals in it, and it's not designed to be a children's toy. So my recommendation was not to use it for that purpose. But a lot of people, you know, just aren't aware of, you know, that so many of these products are chemical intensive and they're designed for a specific purpose. So I think kind of moving generally towards safer chemicals is a really good thing, and whether it's regulatory or legislative or incentive-based, I think there's room for all of these approaches at the table.
Great. Thank you. Another question is, has there been any reaction to the EPA alternatives assessment from the thermal paper industry or the chemical manufacturing industry?
Interesting question. They were all part of it, so there were no surprises there. I think that there are a number of companies that are out there trying to think differently about thermal paper. There are folks that aren't going to move, you know, that they're satisfied with where they are. So you know, there's a range of responses. But I think the good thing is that there are chemicals that are thinking critically about what they're doing. There are companies that are thinking critically about what they're doing and where they're going, and trying to kind of peek around the corner and see if there are different ways of doing things.
Just as a follow-up before you move on to the next question, I would like to point out that the academic community, at least the individuals that I interact with who work on endocrine disruptors, were very pleased with this report. And I think we have to give the EPA a lot of credit for trying to think outside the box, because if the answer here is that there isn't a safer alternative right now, EPA is proposing some creative solutions like electronic receipts. And while the thermal paper industry might not like those solutions or those suggestions, hopefully it will push them to develop safer products. But really, that's a public health protective initiative, and I think they should be applauded for that.
Great. Another
Thanks,
question.
Laura.
Yes, absolutely. Another question here is essentially, you know, what would you recommend, or what would be a way that states, I think, and I assume here that we're talking specifically about states, can promote the development of or access to more data to allow for better decision making in the context of alternatives assessment, substitution, and the like?
this is Cal, and that's a tough one because a lot of agencies have been trying to get more data for a very long time, and I think ultimately, again, these complex issues will take a multi-pronged approach. One of which is, looking at the alternative test strategies that are being developed, and trying to figure out how to use that in decision making that will be protective of human health and the environment. It's going to take a village to achieve that goal. So staying engaged in that. One thing I do want to point out is that I think it was Minnesota. I don't know if any folks from Minnesota are on the line, but they had a project on BPA and thermal paper. So they were working at the state level, looking at how to engage with companies in their state to think more creatively about alternatives to thermal paper. So I think that networking amongst yourselves, and certainly, it helps give the federal government ideas as well. So this cross-pollination is really important.
Great. Thank you, Cal. So I don't see any more questions in the question box, and we are actually at the hour, so time is good here. I want to just, well, first of all, of course, I want to thank both Cal and Laura again very much for excellent presentations today. This was great. Thank you so much. And I also want to just mention to everybody else that this is sort of the last training webinar that the IC2 is going to do before summer. So we're going to take a bit of a break in July and August, and we'll be back in the fall. And I'm just sort of putting this up as a teaser. We're actually working on a webinar even for September, although the details are still a little hazy. But I did want to at least sort of whet your appetites that we are working, as many or most of you know, Ken Geiser has just published his most recent book, which is called Chemicals Without Harm, and we are working on a webinar of Ken's sometime in October to talk about the book, and it will be great. So please do stay tuned, and we will send out more information, in the coming couple of months to give you details about those additional upcoming webinars. So thank you all again for attending, and thanks again to Cal and to Laura for presenting today. And please do let me know if you have any follow-up questions, and take care. Enjoy the summer holidays.
Thank you.
Thanks, Topher. Thanks, Laura. Bye, everyone.
Bye.
Bye, everybody. Thank you so much.